The brain is an organ that serves as the center of the nervous system in all vertebrate and most invertebrate animals. It consists of nervous tissue and is typically located in the head, cephalization, usually near organs for special senses such as vision, hearing, and olfaction. Being the most specialized organ, it is responsible for receiving information from the sensory nervous system, processing that information, thought, cognition, and intelligence, and the coordination of motor control, muscle activity, and endocrine system. While invertebrate brains arise from paired segmental ganglia, each of which is only responsible for the respective body segment of the ventral nerve cord, vertebrate brains develop axially from the midline dorsal nerve cord as a vesicular enlargement at the rostral end of the neural tube. With centralized control over all body segments, all vertebrate brains can be embryonically divided into three parts: the forebrain, prosencephalon, subdivided into telencephalon and diencephalon; midbrain, mesencephalon, and hindbrain, rhombencephalon, subdivided into metencephalon and myelencephalon. The spinal cord, which directly interacts with somatic functions below the head, can be considered a caudal extension of the myelencephalon, enclosed inside the vertebral column. Together, the brain and spinal cord constitute the central nervous system in all vertebrates. In humans, the cerebral cortex contains approximately 16 billion neurons, and the estimated number of neurons in the cerebellum is 70 billion. Each neuron is connected by synapses to several thousand other neurons, typically communicating with one another via cytoplasmic processes known as dendrites and axons. Axons are usually myelinated and carry trains of rapid microelectric signal pulses, called action potentials, to target specific recipient cells in other areas of the brain or distant parts of the body. The prefrontal cortex, which controls executive functions, is particularly well developed in humans. Physiologically, brains exert centralized control over a body's other organs. They act on the rest of the body both by generating patterns of muscle activity and by driving the secretion of chemicals called hormones. This centralized control allows rapid and coordinated responses to changes in the environment. Some basic types of responsiveness, such as reflexes, can be mediated by the spinal cord or peripheral ganglia, but sophisticated purposeful control of behavior based on complex sensory input requires the information integrating capabilities of a centralized brain. The operations of individual brain cells are now understood in considerable detail, but the way they cooperate in ensembles of millions is yet to be solved. Recent models in modern neuroscience treat the brain as a biological computer, very different in mechanism from a digital computer, but similar in the sense that it acquires information from the surrounding world, stores it, and processes it in a variety of ways. This article compares the properties of brains across the entire range of animal species, with the greatest attention to vertebrates. It deals with the human brain insofar as it shares the properties of other brains. The ways in which the human brain differs from other brains are covered in the Human Brain article. Several topics that might be covered here are instead covered there because much more can be said about them in a human context. The most important that are covered in the Human Brain article are brain disease and the effects of brain damage. The shape and size of the brain varies greatly between species, and identifying common features is often difficult. Nevertheless, there are a number of principles of brain architecture that apply across a wide range of species. Some aspects of brain structure are common to almost the entire range of animal species. Others distinguish advanced brains from more primitive ones, or distinguish vertebrates from invertebrates. The simplest way to gain information about brain anatomy is by visual inspection, but many more sophisticated techniques have been developed. Brain tissue in its natural state is too soft to work with, but it can be hardened by immersion in alcohol or other fixatives, and then sliced apart for examination of the interior. Visually, the interior of the brain consists of areas of so-called gray matter, with a dark color, separated by areas of white matter, with a lighter color. Further information can be gained by staining slices of brain tissue with a variety of chemicals that bring out areas where specific types of molecules are present in high concentrations. It is also possible to examine the microstructure of brain tissue using a microscope. and to trace the pattern of connections from one brain area to another. The brains of all species are composed primarily of two broad classes of brain cells: neurons and glial cells. Glial cells, also known as glia or neuroglia, come in several types and perform a number of critical functions, including structural support, metabolic support, insulation, and guidance of development. Neurons, however, are usually considered the most important cells in the brain. In humans, the cerebral cortex contains approximately 16 billion neurons, and the estimated number of neurons in the cerebellum is 70 billion. Each neuron is connected by synapses to several thousand other neurons. The property that makes neurons unique is their ability to send signals to specific target cells, sometimes over long distances. They send these signals by means of an axon, which is a thin protoplasmic fiber that extends from the cell body and projects, usually with numerous branches, to other areas, sometimes nearby, sometimes in distant parts of the brain or body. The length of an axon can be extraordinary. For example, if a pyramidal cell, an excitatory neuron, of the cerebral cortex were magnified so that its cell body became the size of a human body, its axon, equally magnified, would become a cable a few centimeters in diameter, extending more than a kilometer. These axons transmit signals in the form of electrochemical pulses, called action potentials, which last less than 1,000th of a second and travel along the axon at speeds of 100 meters per second. Some neurons emit action potentials constantly, at rates of 100 per second, usually in irregular patterns. Other neurons are quiet most of the time, but occasionally emit a burst of action potentials. Axons transmit signals to other neurons by means of specialized junctions called synapses. A single axon may make as many as several thousand synaptic connections with other cells. When an action potential, traveling along an axon, arrives at a synapse, it causes a chemical called a neurotransmitter to be released. The neurotransmitter binds to receptor molecules in the membrane of the target cell. Synapses are the key functional elements of the brain. The essential function of the brain is cell-to-cell communication, and synapses are the points at which communication occurs. The human brain has been estimated to contain approximately 100 trillion synapses. Even the brain of a fruit fly contains several million. The functions of these synapses are very diverse. Some are excitatory, exciting the target cell. Others are inhibitory. Others work by activating second messenger systems that change the internal chemistry of their target cells in complex ways. A large number of synapses are dynamically modifiable. That is, they are capable of changing strength in a way that is controlled by the patterns of signals that pass through them. It is widely believed that activity-dependent modification of synapses is the brain's primary mechanism for learning and memory. Most of the space in the brain is taken up by axons, which are often bundled together in what are called nerve fiber tracts. A myelinated axon is wrapped in a fatty insulating sheath of myelin, which serves to greatly increase the speed of signal propagation. There are also unmyelinated axons. Myelin is white, making parts of the brain filled exclusively with nerve fibers appear as light-colored white matter, in contrast to the darker-colored gray matter that marks areas with high densities of neuron cell bodies.